Hello, thank you for having me. So thank you to Wayne and to Richard for the invite and for everyone here and online for coming. My name is Kylie and I'm going to be talking today about a project that I've been working on for the last year and a bit around a little fungus that I found actually with QMS on a foray back in 2023. And I'd like to start as well with an acknowledgement of country. I would particularly like to acknowledge the traditional owners of the lands on which I have done most of this work, particularly the Yugambeh people, which is where the mushrooms I'm going to be talking about today have come from. I'd like to recognise the fact that there are First Nations and traditional owners were the first scientists in Australia, and that they continue their cultural, spiritual and scientific connection to country today. And I recognise their valuable contributions to Australian and global society. So a little bit about the research that I do. My research in fungi has spanned for the last now decade since I started my PhD back in 2015 when I was working on a drug design project around invasive fungal infections. So my background is in synthetic organic chemistry, and I use that to explore the world of medicinal chemistry, which is a very long-winded way of saying that I make drugs for a living and I'm allowed to. More recently, I've been looking a bit more into native mushrooms as a source for medicinal compounds. And so this work started in about 2022 with some work that I was doing with Little Acre mushrooms, which is a grower here in Brisbane, and some of the hericium that they were growing and interested in learning a little bit about. And the project has really expanded out into a lot of different species of mushrooms now. But what I'm really interested in is having a look at the medicinal compounds that they make, and also having a look at characterizing some of their molecular features, their genome sequencing, their metabolomes, the things that they make, the proteins that they make, and how they make some of these compounds as well. Underpinning a lot of the medicinal work that I do in native mushrooms is the fact that in the last 4 decades, around about 70% of commercial drugs that were approved since 1981 or 1989, I think, yeah, 1981 through to 2019, over 70% of those are either a natural product, so they come directly from some sort of natural source, or they are derived in some way from a natural source, be that semi-synthetic, or they are used to mimic the structures of a natural product or a biological molecule or something along those lines. And the reason for this is really because nature is really good at making a huge diversity of compounds that inherently have biological activity that we can use to treat diseases and various other things. But there is a question then when we're talking about things from natural sources, and we're looking at drugs, which are more often than not single individual isolated compounds of, firstly, I mean, how do we even find these things? We start from a plant, a mushroom, whatever that happens to be, but how do we even know what we're looking for or what we're looking for? And then secondly, how do we know what biological activity to screen for when we're thinking about these big or small complex organisms that make thousands and thousands of different chemicals? How do we know what we're actually going to be screening for? And this is fundamentally what I'm going to be talking about today. So I'm going to start just by introducing a bit about fungi as medicine and how we use it as medicine, and how it has been used in medicinal chemistry and in drug design projects. And then looking at how we find bioactive molecules in fungi. So answering those 2 questions I mentioned before from the perspective of a fungus that I collected in 2023, the Kretzschmari that I mentioned in the first slide, and looking at some of the antimicrobial compounds that we've been finding in them over the last year. I mean, fungi need no introduction to this audience, but we've been using mushrooms and fungi for millennia for medicine, for food. More recently, we've had applications like bioremediation and plastic waste removal. They are a hugely diverse group of organisms. We have, I think, millions of different species worldwide, most of which we know nothing about. We don't have names for most of them. And in Australia, that is most certainly the case. And because they occupy quite a lot of different ecological niches, they do a lot of different things. They live practically everywhere, even in space. They can make a lot of really highly specialized molecules that can do a lot of different things, which makes them a really incredible resource for bioactive molecules. We have quite a lot of drugs already that are commercially available that have come from fungi. Some of the more famous ones include things like penicillin, which most of you will be aware of, but then also things like, if I can get my laser pointer, this here, which is echinocandin B, which is an antifungal drug used to treat invasive fungal infections, and was the inspiration for one of the 4 or 5 classes that are used to treat invasive fungal infections. And this comes from an Aspergillus species. And then more recent examples like this one down here. We also have a lot of immunosuppressants that come from fungi as well. So cyclosporin as an example, which some of you may or may not have heard of. And a few of so the 2 examples I have here actually come from entomopathogenic fungi, and then another one here that comes from a penicillium species. I think this one is used for, actually I can't remember what that one is used for, but these are all immunosuppressants anyway, mostly for people who have undergone organ transplant and that sort of thing. And we have lots more. So statin drugs is one of the more famous examples of a commercial drug that comes from fungus that we use today. That started with lavastatin, and that comes from, again, another microfungus, an Aspergillus species, and then a couple of other ones that used medicinally or recreationally, depending on how you feel. So what about Australian macrofungi? What do we know about the medicinal compounds that come from them? And not a lot is the honest answer about that. There is some work in marine microfungi that is done in Australia. There's quite a lot of that actually. And a few other species of microfungi from soil, for example, at UQ. There's a few groups that work on soil fungus and marine fungus, and there's quite a lot of bioactive compounds that come from those that have been found. But in terms of our mushroom species, so our macrofungi, there's not really a lot in the literature that you'll find around these. So I've just given a couple examples here. This first one, which is from a hyphiloma, this compound which this group isolated from the hyphiloma, this phasculuric acid C, which they had found, at least reported in the paper, was toxic for human cervical cancer cells, not to non-cancer, not to normal human cell lines, which is not common. Often when things are cytotoxic, they're cytotoxic to everything, which is so the fact that this is selectively active against cancer cells is quite impressive. And this is actually work that has come out of some of the research that I've done in the last couple of years, but not something I'm talking about here today, from our native Hericium species. You might know its cousin Hericium erinaceus, which is lion's mane. And Hericium is very well known as producing compounds that have really amazing effects on axon growth in neurons and cognitive function. And in our native species, we have done some work with the Queensland Brain Institute at UQ, looking at whether or not our native species can do the same thing, and it turns out that they can. And so I've just given a very quick example of what that looks like. So this little image down the bottom here is, these are hippocampal neurons that were treated with just the solvent that we use to dissolve these things up. And if you compare that then to this image of a neuron here, after being treated with the chloroform extract from one of our Hericium specimens that we have, you can see that the axons in these neurons are absolutely massive compared to if they're not treated with anything. And so the talk today is really about antimicrobial compounds. My background comes from infectious disease drug design, and that is where my interest primarily lies in my other research as well. And so there's been a little bit done around this in Australia and in Australian species. This first example that I came across is actually a very recent one from 2025. The majority of the authors on this paper actually aren't in Australia, other than Tom, who most people in the audience would probably know. He's the mycologist down at the Royal Botanic Gardens in Victoria. And so this paper is one of many actually on these photoactive anthraquinones from Quaternarius species that have activity against different species of bacteria, in particular Staphylococcus aureus, once they are activated by light. Oh, too far. And this is a slightly earlier example. This actually comes from UQ. And it's a There's a lot of numbers and you probably can't read any of them, and that's fine, you don't have to. This paper essentially was looking at the antimicrobial properties of 12 different basidiomycetes that were collected from around Queensland. And the crux of this is that we have a few species here that have, so there's a Hohenbuehlia, Bromeria, Streptomyces, and a few other species, Cantharellus, Amanita, that had quite impressive activity as just the extracts. So these aren't isolated compounds against some gram-positive and gram-negative pathogenic bacteria and also some pathogenic fungi as well. The compounds from these weren't identified, and so there's not actually a lot of work other than the stuff on the Quaternarius, and there's a few other papers as well on antimicrobial properties of the larger fungi that occur in Australia. And that's really what my work has been interested in, partly at least in the last couple of years after the work on Hericium. And this is another big complicated slide with a lot of information, but this is just to summarize the way that my research works and how we're able to look for these sorts of things. And really what my work tries to do is integrate a couple of different approaches for looking at these molecules. And I'm only really going to be talking today about this bottom half, which is the more traditional approach to looking for these biologically active molecules, where you take some sort of natural source, a mushroom, obviously in this case, and you screen it against something and you pick out the things that are active, and eventually you come away with something that is both active and pure. The very start of this work is where my interaction with the lovely people of QMS and various other amateur mycologists, citizen scientists, and other organizations like Queensland Herbarium and Nigel and Tom comes from. This is probably my favorite part of the work outside of the actual chemistry of it, which is being able to go out and talk to people who really love mushrooms and hear what they have to say about what is interesting to look at, because quite frankly, 3 years ago when I started this, I didn't know anything. And it was through coming to these sorts of meetings and interacting with people and just asking questions and engaging with websites like iNaturalist that you really come to understand what might actually be interesting to look at, because it's all well and good going out and picking a mushroom and just testing it against some random thing. But you're spending your time a lot more smartly if you're actually talking to people who know about the front end of that stuff and have some idea of where there needs to be more research. And so I've learned a lot in the last couple of years about how to do this sort of thing properly. So when I'm making these collections, all of the mushrooms that I work with, I collect myself. And so with that has come a journey of learning how to take proper field notes and proper photos and what to look for. In terms of characterizing these things, because a lot of my work also feeds into looking at the biodiversity of fungi in Australia as well. Since I'm gathering all of this information and I'm out there collecting, you may as well. And so with that also comes the microscopic characterization, and that has been another great learning curve over the last couple of years. I knew how to use a microscope, but knowing what to look for in fungi is a whole other thing. And learning how to culture them was also a journey in mold, mostly. Isolating a lot of mold, but also isolating a lot of quite interesting things like this little Bolitaceae species that I have up in the corner that I was able to isolate out last year, and some of the Hericium. I have a nice big collection of Hericium from Tasmania and Queensland currently sitting in the fridge. That I've been able to isolate out, and it's no small task if you've ever had to isolate these things, particularly basidiomycetes that just don't like to grow when you have ascos to compete with. And then, so essentially what I can then do with these, and part of the reason I take living cultures, not only because the DNA is better when you have these nice living cultures that you can then grow up when you want, is you can also grow them up and make extracts from them, because the mycelia of fungus will frequently make different things to what the fruiting bodies do. And so I use the live cultures for that, as well as the DNA part of it. And then, I think we'll cancel that. And then the field specimens, obviously, I use for the extraction part of the research and also to help with species ID. Anything I collect, I also collect enough to voucher with the herbarium as well. Yeah, there we go. Okay, All right, so to our first question of how do we find these bioactive molecules? And there's a few different ways, and I'm only going to talk about one of these, which is our bioactivity guided purification. And I went over a little bit about that before. And essentially, you make extracts of your mushroom, you screen them against a model of disease, you take the active ones, you purify them more, you test them again. One day you'll get something that is pure. And this is really what we do with the mushrooms as well. So we dry them, we have thousands of chemicals, we test them, we pick out the active ones, we purify them again. We have maybe tens or hundreds of compounds, we test them again, and so on and so forth. And you can imagine it takes a long time to get anything out. To be honest, and if there are any people who do natural products chemistry in this audience at all, I will say that fungi are much more forgiving than plants. As you'll see in a minute, and I'll provide a little bit more context for that, simply because of how much you get out of them compared to plants. So it is very labor intensive, but you're more likely to get things you can actually work with. And there are other issues as well. One of the big ones, of course, is that you do still get very limited amounts of material. And some mushrooms, of course, are tiny. So, I mean, you're only ever going to be able to get out so much at the end of the day. So you can't really screen for everything, which leads us to our next question of how do we then know what we're actually screening for? There's however many diseases out there, and we don't know what a given compound is going to do just by looking at it. You have to test it. And there's, again, a few different ways. So one is just the random testing. You can do whatever's convenient, and I have certainly done that. I know how to do all of the antimicrobial testing myself, so might as well while I'm there. There's also, if you can identify either what the fungus is, or you can identify what the compounds are that it's making, we have the benefit of the last 100 years or so of literature that we can potentially look up to see what maybe things that have been done before. There's also traditional medicine systems. So there is a history of indigenous use of fungi in Australia. It is very, very poorly documented, but it is there. And of course, around the world, mushrooms have been used a lot more heavily in traditional medicine systems, particularly in Asia. And then this last one, which is probably the one that I'm the most interested in, which is the use of chemical ecology and field observations and getting out there and actually looking at these things and how that can be used to tell us a bit about what these things do. So what is chemical ecology? So the definition up there is essentially what we're looking at is how an organism relates to its environment and its surroundings through chemistry, essentially. And this little picture that I've included here is actually a picture I took myself on a collecting trip I did last year in North Burnett and Barracuda State Forest. And this was in Barracuda. It's a little bolete that I found one morning while I was walking. And what you'll notice about this bolete is that it is absolutely covered in mosquitoes right underneath on the pore surface there. And it was only this one. There were a few of these around. This one was a slightly younger one, and the mosquitoes seemed to be going absolutely crazy for it. And it's observations like this that can be really useful, because you can look at this and you immediately think, well, they must be going there for something. And what that could mean is that they're producing an attractant that is attracting those mosquitoes to it. And how could that be useful? We can use that as lures, for example, if there is something in there that we could use for mosquito traps. So things like that, like simple observations like that, can be super, super helpful when we're trying to think about how we might want to test this. And I didn't sadly make a collection of this because my permit was not for boletes at the time, but I know where they are, so I'll be back. And yeah, so fundamentally, if we understand why these organisms, why these fungi are making the things that they are making, and we have some idea of how they're using those to interact with the world around them, then we can use that to help triage what tests we might like to do. So at the end of these sorts of processes, you're often only left with like a milligram or so of material, which is quite tiny. So if we think back now to our antimicrobials and why our fungi might want to make some of these molecules, and the antimicrobials in particular, it's important to think, of course, that mushrooms aren't just mushrooms. They are a whole lot of mycelia underneath the ground, or whatever their substrate happens to be. And under the ground or in their substrate are other things, bacteria, insects, fungi, other animals as well, that they have to compete against, and they have to fight against for resources and for space, and to fight against in the case of pathogenic bacteria and fungi. And then above the ground, we have yet more animals and fungi, sun, rain, bacteria, insects again, who all might want to eat or in some ways destroy this little mushroom. And maybe the mushroom wants to be eaten by an insect. Some of them do, but maybe they don't. And so when we're thinking about those sorts of interactions, it can really help us figure out what they might be doing that's interesting if we're looking at them. An example of that is actually in the Hericium mushrooms that I work with. Their primary defense is chemical defense. And the Hericium mushrooms that I work with, one thing that you'll notice if you've ever seen them in the field is that they very rarely, if ever, have mold on them. And if I have gone through all of the observations on iNaturalist in Australia, and there's 300 odd, 350, I think at the moment, observations of Hericium in Australia. And of those 350 to 400 or however many there are only 6 that you'll see that have mold on them. And they last in the environment for a really long time. And so that gives us a hint of it. Maybe they're making antifungal compounds or other antimicrobial compounds. And if you look in the literature, there's actually not a lot of information on that sort of thing. But I can tell you from my work that they do actually make antifungal compounds as well. On to Kretzschmari, which is what I am talking about today. So Kretzschmari is an ascomycete fungus that some of you might have seen without actually realizing it. It tends to look pretty cryptic. This is actually one of the more advanced stages of the fruiting bodies that you'll see. They have this very smooth, almost velvety gray surface. And when they're really young, they form basically these small gray discs on the side of trees. They're parasitic, so they around the world, they are very significant pathogens, particularly of macadamia. Australian species, as far as I know, well, it is undescribed. I think a little while ago it was given the name Kretzschmari Agnew-Francis, which is the Northern Hemisphere species, and it does bear a lot of morphological similarities to that. When you take these cross sections of it, you'll see, and so when we collect, this is actually the specimen that we collected that I've done a lot of this work on, has these really nice concentric bands, which is very similar to Daldinia, I believe, and other species like that. We couldn't actually identify this when we first found it on this foray. It took a couple of months for me to figure out what it was. In terms of what they make, despite the fact that they're really, really prevalent, they're all over the world, and they're a huge pest for certain crops. There's been almost nothing done in terms of research into the species in the whole entire genus, and they're a pretty big genus. I think there's somewhere in the region of about 36 to 40 species in the Kretzschmari genus. There are only 2, I think, reports. I've only got 1 up here, but I've since found 1 more of the sorts of metabolites that they make. So screening some of their natural products. And this wasn't, as a natural products chemist, what I would really call a natural products chemistry paper. Really, all they were doing was just having a look at the mass spec and a big mixture of things, which isn't a particularly thorough way of actually trying to identify anything. They found 1 compound in this particular study in very, very minor amounts, and that is the extent of everything that's ever been done. And Australian species are not investigated at all. In fact, you'll see nothing about them in the research, at least as far as I know. I mentioned that the one that I have done a lot of work on the last year or so was one that was collected on a QMS foray. And this was in Binna Burra back in June of 2023. And so this was actually right at the end of the foray. And I remember we were coming back. I think Richard and I had massively overshot where everyone else was, and we were making our way slowly back, and Wayne was looking at something else. And I was just wandering around and almost went face first into a stinging tree. But under the stinging tree was this. And I didn't know what it was, but it obviously, I mean, very clearly looked fungal. And consulting other people who also happened to be there, no one seemed to really know what it was. And I was interested, so I asked if I could take it back with me. And I was very graciously allowed to take it back. And so we took some of the fruiting body. And so this was work that started off with a project student later that year, over the summer of 2023, 2024. And he took some of the fruiting body that we collected, and he milled that, and it makes like this, it looks like charcoal, basically, just charcoal powder. And we got about 30 grams of this dry powder, and he took some of that, but not all of it, and we separated it into 3 lots, and we extracted it in 3 different ways. And from this 8 to 9 grams of dry powder, we ended up with 1.5 to 2.5 grams worth of extract dried down after vacuum. And if you were a plant natural product chemist, like that sort of return on investment is practically unheard of. You'd be lucky to get from 8 grams of a plant, you'd be lucky to get 50 milligrams back of anything. So I love giving this talk to natural products chemists because it makes them very jealous. And I hope to encourage more of them to turn to mushrooms because you'll be much happier. So after my project student finished up, we took some of this chloroform, one of the extracts we made, which was a chloroform extract, and we use these different solvents because they take out compounds with different polarities, and so we want to get a nice cross section of the sorts of things that are being made. And so this chloroform extract, we use a technique called nuclear magnetic resonance. Some of you might know of MRI. It's the same thing, but smaller. And we can use that to look at what sort of compounds are being made. And so we use NMR routinely in this sort of chemistry to identify and elucidate structures if they are known or not known to us. And we can tell a few things from this, mainly that we have a lot of compounds called which have functional groups called aldehydes in them, and we can tell that from these little peaks here. And I won't spend too much time talking about the science of all of this. I don't particularly understand the physics of it myself. NMR to me is sort of a tea leaf reading exercise, but you do get very good at reading the tea leaves after a while. And so we can tell a couple of more things about these. They have these aldehydes, they're highly oxygenated. We also have some fatty acids that seem to be attached to these in some way. And from there, we took that, and a new student, a PhD student who was trying not to write her PhD thesis and doing a fantastic job of it, took this extract, and she took some of the fractions that we made. So we separate these using a chromatographic technique, which separates things again based on their polarity. And she took some of these fractions that we had made and pulled together. And so we're talking about, I think, 8 different fractions that she was working with. And from those 8 different fractions, we were able to elucidate, I think we're up to eight different compounds now. And all of these compounds that we've been able to isolate have this marasmane core. So this is a type of sesquiterpene. And so all of them have this little marasmane core in the middle here, and attached to those are a whole bunch of oxygens and a little fatty ester that we could see in our NMR. And in that NMR, the other thing that we could also tell was that actually there were only a couple of compounds in there. The things that they seem to be making seem to be mostly two or three things. And there is some literature on these. So obviously from the name, the first thing that the marasmanes were actually isolated from was a marasmias. But most of them since have been isolated from Lactarius and Brucella species. And when I say most of, I'm really only talking about a very, very small handful of things, because actually these types of compounds are incredibly rare. And the only examples of them that are known are not actually synthesized by the mushrooms all the time. They're actually only made in response, it turns out, to injury. And something else that was very interesting when we started looking in the literature on these was that none of them have ever been found in ascomycetes at all. They're exclusively found in basidiomycetes. So the fact that we found so many of them in an ascomycete was pretty interesting. So as I said, these compounds were found in the literature, and this is literature dating from the 80s, 90s, and like very early 2000s. And what they found, what they were able to determine after a while was that these mushrooms, and particularly the Lactarius and the Russula, when you injure them and you break them and they start producing that latex, before you injure them, they have these what are called these volutinal fatty esters, and these are fairly tasteless compounds. They don't really do much. They're not cytotoxic. They're not active against anything really. But when you break them open and they start making these compounds pretty much instantly. And the end products of these, which include those marasmane esters along with a few other things, are incredibly antimicrobial. They're cytotoxic, and they also have anti-fedant activity and insecticidal activity as well. And they were able to tell this. It was actually really cool. They got the mushrooms intact, so they very carefully collected them, and they extracted them whole. And they also took some and they ground them up, and they extracted them at different time points after having ground them up. And they were able to find, as you would expect after what I just said, that when they extract the mushroom whole after 0 grinding time, you find basically just these fatty esters that do nothing. And then after a while, after having ground it up, eventually it makes these compounds here that have all of this wonderful activity. So these are obviously a defense compound then to protect against feeding from things that they don't want to eat them. So we looked at that and we thought, well, maybe some of our compounds do that as well. And so there's a couple of groups over just over the road from where I am who do things with insects, and they look at insecticidal activity of natural products. And they do these really bizarre assays with Drosophila where they can inject individual fruit flies or feed them orally, which seems much easier. But this was by injection. I don't know how they do it, but they injected these poor little fruit flies with these extracts. And it turned out anyway that they weren't very active. They showed very, very minimal to maybe moderate activity against these insects. And I don't have access to opossums, so we didn't really look at the anti-feedant activity of these things. But I mean, when you think about it, this mushroom doesn't have a lot of water content. It's basically charcoal. I don't know who would want to eat it anyway. Maybe an insect, but certainly I can't imagine any possums or anything would want to be eating this thing. So we're now left thinking about what else can it do. And so I actually ended up going back to a different spot where I knew that this fungus shows up, because I was interested still in knowing if, like the Russula and the Lactarias, they made these compounds all the time, or if they made them just when you injured them, which didn't seem like they would, but might as well figure it out. And so I went to Tamborine. And I had already collected one specimen of this in August of last year. And so this picture here is the one that I collected in August, I think, or is it this one? No, it's this one from August. And then in January of this year, I collected it again. And the fruiting body was still there. This is the same fruiting body on the same tree. And you can actually see, oh no, sorry, I was right. This one is the one that was from August last year. This is from January, because you can see the little cut marks from where I took away from it last year. So exactly the same fruiting body. And when I came back in January, I actually came with a bottle of solvent, and I grabbed some of it and I threw it straight into the solvent. And I wanted to look, I then took that solvent, dried it down, and had a look at using NMR again to see what it was making. And the long and short of this is that you see basically the same things. So these, the Kretzschmari seem to be making these all the time. And the fact that the fruiting bodies exist for months and months at a time, and this thing is still there even now doing its thing. The tree doesn't look as good, but the mushroom looks fine. Suggests that these probably have antimicrobial properties. And so that's what we looked at next, particularly at the antibacterial and antifungal activity. So we're looking at human fungal pathogens and gram-positive and gram-negative bacteria using just very quick and easy cell-based assays. So again, a lot of numbers. You don't need to be able to read them. But what was really interesting about these numbers, so we have this top one here, this K1, is one of the compounds that we isolated. So this compound that's kind of in the background here is this K1, and we isolated this from one fraction from the silica column, which was a fraction of a fraction of a fraction. 16 milligrams of that is wild numbers for anything. So we did all the tests on it because we could. There we go. Yes, and so we took that compound, and I've only shown data for one of the extracts. This is the chloroform extract, but the methanol one showed more or less the same thing. The water extract showed basically nothing. But what was interesting was that particularly against our fungus, the organic extracts, the chloroform and the methanol extracts, both showed this really, really strong activity against the species that we tested. And so this is Candida auris and Candida albicans. These are two of the species that are listed as fungal pathogens of top critical priority concern by the WHO. These two up here are our controls, and so these are drugs that are used as standards for treatment of invasive fungal infections, and so we use that obviously just as a benchmark to compare against. And the numbers maybe don't look as impressive, 14 points, so 15 microgram per mil compared to less than a quarter of a microgram per mil. But I mean, you think about the fact that this is an extract, there's multiple compounds in there. And what was really remarkable So this number here, which is our MFC, this is minimum fungicidal concentration. So when we're doing these assays, what we're looking at is what is the lowest concentration that will completely kill the fungus. And that's what these concentrations represent. And when you have that concentration, you can then take little bits out of the wells that you do these in. These are done in 96 well plates. And you can spot those. You can then take it away from the drug and see if the fungus grows back. Because what often happens, and this is the same for antibacterial activity as well, is that some drugs will completely kill a fungus and some drugs will just stop it from growing. And obviously, if it stops it from growing, the moment you stop treatment, it's just going to come back. And this compound, which is one of the major components, is the major component of those extracts. While it did have some activity against our species here, so particularly our Candida auris, it had absolutely no fungicidal activity, whereas our extracts were completely fungicidal all the way up until their MIC concentration, which is not common at all. There we go. Okay. And antibacterial activity, so the assay is very much the same. So we have these little plates, we inoculate it with our bacteria, and we look at where what the lowest concentration is that will completely kill the bacteria. On the left is our gram-positive bacteria, and on the right here is our gram-negative bacteria, which are so they're all. We have two different Staphylococcus aureus. One of them is drug resistant. And both our organic extract and our isolated compound do have some activity against the drug-resistant and the regular strain of our Staph aureus, and they have some activity against other gram-positives, but not so much the gram-negatives. But still, the fact that they have activity against the gram-positives is still quite a promising sign. And we're yet to figure out exactly what it is, other than if there's other things other than this compound that's doing this part yet. We've also looked at the toxicity of these, because quite often what happens, and as is reported in literature around some of the other marasmanes, they also are toxic to human cells, and they can sometimes be toxic to blood as well. So this hemolysis is looking at the toxicity to red blood cells, which are obtained through through hospitals. So these are patient samples, and it's looking at how much what percentage of those blood cells are completely exploded or lysed by the extracts, and the cytotoxicity, which is looking at the toxicity against a human cell line, so HEC293, which are kidney cells. And so at our top concentration, you see complete inhibition, which is not completely unsurprising. It was pretty high concentrations at the top end there. But the IC50, which is our 50% concentration value, the concentration that inhibits 50% of the cells, and that's the common sort of value that we're looking at. It turns out that number is actually not too bad. So medium to like low cytotoxicity, which is promising. And again, these are just human cell lines. These are in vitro assays, but almost no hemolysis, which is great. So your blood's safe. Okay. And so that's sort of where we were at actually with the Kretzschmari stuff. So we found some really cool compounds that these things are making, and they seem to have some really interesting activities. We haven't yet figured out what the antifungal compounds are that are giving those extracts a really, really strong fungicidal activity. We're hoping to be able to do that in the next sort of few months as we're finishing up some of that work. But just to summarize, the Kretzschmari is actually just one of a million different mushrooms that I'm actually looking at the moment for antifungal and antibacterial activity. So I also do work with Romaria and more recently with bolets, because I just keep finding them. I tend not to work with gilled things, but the corals and the bolets, they're all in there. And the really big takeaway from this is that fungi, and native fungi in particular, have a lot to offer in this space. And being able to do the research in the chemistry world and then crossing over into things that are absolutely not my area, but I do an okay job of pretending, has been a lot of fun. And I would encourage anyone to try the same thing if you are a researcher. I'm currently also doing some work through Bioplatforms Australia, which has helped to fund a lot of the genome sequencing and all of that sort of stuff that we're doing. And so that work, as well as helping me in the natural product space, also has the side benefit then of being able to really dig down into some of the biological diversity side of things, and there are some researchers at UQ who are working on some programs for using genome sequencing data to map out taxonomy of various species. And so I'm working with those groups, with particularly Mycological, because I have so many of those. And so we're still doing some more extraction of those and about 1 million other different mushrooms at the moment. So more to come in that space. And it just remains for me to thank everybody here and to thank my very, very long list of supporters and people who have helped me along the way. And this list is, I think, even though I only did this talk a couple of months ago, already out of date again. So a lot of the work was done by these first few people here. So Laura, Adan, Janavi, Holly. So they were the students who have sort of powered this along. And I've also received quite a lot of help from QMS, obviously, who helped me with the collection of this species, the herbarium, and Nigel and Tom as well on this and other projects. And a lot of this was done through funding that came from the Australasian Mycological Society and through the Royal Society of Queensland, as well as UQ as well. And so that is me done. It's a lovely, this is actually the foray where we collected the mushrooms. I hope you don't mind that I put it up, but yeah. Yeah, so thank you everybody for listening, and I'm happy to take questions. I'll repeat them. All stunned into silence. Yes. Yes, so Wayne was asking if the extracts that we found were lethal for Candida auris, and yes, they were completely. So the chloroform and the methanol extracts were both, we kept going lower because normally when you're testing extracts, you go at a high concentration because you're not expecting there to be a heap of activity. There's just too many things going on. But they were completely active, and so we had to keep going lower and lower and lower into now the realms of the sort of activity that you see from standard drugs. So these extracts are as active as some of the standard drugs that are used to treat these infections. Yeah. Yes, Alice. Mhm. Yes, so Alice is asking if there are ways for us to essentially simulate or improve the yields of some of these bioactive compounds by simulating different conditions. The short answer is yes. The long answer is that it's very complicated because obviously like they're responding to different things and you don't know what that is. As particularly for the antimicrobial compounds, a lot of the time, so a lot of these studies that are look at these sorts of things are done using lab-grown mushrooms, so in sterile environments, or mycelia, and the mycelia produce very, very different things to the fruiting bodies, not better or worse. Like the mycelia can be a great resource, and that's part of why I take these cultures as well. But if they're not being challenged in the same way, they're not making the same things, and it's difficult, I guess, if you're working with antimicrobials, because you have to know what they're trying to fight against, I guess, some of the time. Sometimes they make these things all the time, but a lot of the time they need that thing there. And mycorrhizal species, I mean, it's anyone's guess. Yeah, yes. Thank you. Yeah. Yeah, so John generally was asking me if I have how it is that I get the bolates into culture, and I will say I can't get all of them into culture, just some of them. Some of them seem to like it and some of them don't. I guess the question really is what they're actually getting from their mycorrhizal partner, because sometimes it's things that you can give them. There are like chanterelles and stuff that you can culture. The meter I use for the bolates depends. I normally, if I'm taking from the field samples, I'm normally culturing on a few different things at once just to cover my bases. So I use just your standard potato dextrose agar, always water agar. These are all with antibacterials in them. And then there are two other one media I use mostly for wood rots, which uses 2 antifungals in addition to, which is a BOP media, I think it's called, or I think one of the names is Ben 10, which I really like because this was a media that was described first, I think, in the 50s, obviously well before Ben 10. But anyway, and MMN or MMN media, melons, some, I can't remember it's a big long name. If you look it up, it's very findable. But that one uses malt extract and a few other additional thiamine and a few other things. And there's another one I use for chanterelles as well, in addition to that, which is the modified fries agar. Yeah, and then you just have to watch them intensely for a week. Okay, yeah. So question in the chat is, if you were to find a useful commercial compound, would you be needing to grow the fungi to extract more, or would you then be making a synthetic version? That is a great question. So obviously mushrooms, very ephemeral. They grow very transiently. If you've been through Southeast Queensland over the last week, one day they're there and the next we've had like a day of sun and they're gone. So it's difficult. If you're trying to doing the bioprospecting and bioharvesting for drugs is not the best way to go about it because you need a lot. And mushrooms, I mean, some mushrooms grow easy and some mushrooms don't. And yes, if you're finding something, for example, in a romaria, I mean, good luck. That's you're not culturing that. So one way to do it is through total synthesis, which is where you identified the compound and you make it synthetically. More often than not, though, the things that you actually isolate from the fungus or from wherever are not directly usable as drugs. Something that is active is not something that is druggable. So you have to do a lot of other medicinal chemistry to these things more often than not to actually get them to a point where you can use them as a medicine, because there's so many other things that go on. They have to go through your body to where they need to go and not do other things, which is hard. And, mushrooms aren't making things for us, they're making things for them. So the other way that you can do it is if you know how it's making it, and you can take that and put that into a bacteria or a yeast or something that you can then ferment and grow lots of easily, you can do it that way. That technology is mostly there, but it's still expensive and you still need to do a lot of research first to get to that point. But no, I don't think even if you were to find the next best drug in a mushroom, you wouldn't then be using the mushrooms as the source of that drug. Oatmeal agar. Haven't tried it. I use malt and PDA. They seem to do all right, but oatmeal's a good one. I quite like, for ascos, that's good. The Kretzschmari I haven't been able to get into culture because when I first collected it, I didn't know what it was and I wasn't paying attention to the right bits. But now that I do know what I'm looking at and where the things are that I need to get, I'm going back for more because I don't have any stuff that hasn't been freeze-dried yet. Okay, so the question was, are the concentrations of the compounds in the mushroom the same versus in the mycelium? And no, usually not. And oftentimes the mushroom will make totally different things to the mycelium. Yeah. C is carbon. Yes, yes. So sometimes you draw them. Organic chemists lazy by trade. And so the less we can draw, the better, which is why you usually see us just drawing lines, hieroglyphics, I guess. Sometimes we draw the C's and sometimes, most of the time, we don't. But yeah, it'll be carbon. Is there more? Okay, there's another question from the chat, which is, when you identify and find a commercial use for the mosquito attracting compound, can I buy shares in your spin out company? Sure. I have to go back and get it first. Is there any other questions? Yeah, yes. Mm-hmm. I'll see if I can get back to the slide. So the question was so one of the examples I gave where they had, of course, they had some compounds that they isolated from a mushroom that were active against, selectively active against cancer cells. And has anything been done on that? And I've spent a lot of time getting to the slide just to tell you no. Yes, yes. Look, what often happens in these things is that for people who are doing natural products chemistry, there's kind of a few different ways that you might approach it, and one of the ways is um, this is also true for medicinal chemistry as well. There are a lot of old school chemists who would much rather just be doing chemistry for chemistry's sake, but you can't get funding for that, and you can't get published for that. So they'll tack on other things, and one of those things is quite often just very easy tests that they have access to, which is where the random testing and whatever you've got access to comes into it. And I'm not saying that that is what happened here. In fact, knowing those researchers, that's almost certainly not what happened there. But the it's a lot of work, and I can't remember how much of this compound they actually got out of it. The other thing as well, as I mentioned before, that a compound that is active is not necessarily a compound that is druggable, and having that activity data is an important, very important first step. And having the toxicity against non-cancer cell lines is also a very important first step, but there's a lot of other stuff that goes into that, and so that doesn't always get published. And so quite possibly what has happened is that they have done some of those other assays and it turns out it's just rubbish. It might be toxic in other ways, or they might have done animal studies and it just didn't do anything. Yes. Okay. Yeah, what machine am I using when I do the extractions? I don't know if I actually put a picture of. Oh, do you mean this thing? Yeah, so that is well, that's not the extractions, that's just the freeze dryer. So that's what I use to dry it rather than just a regular dehydrator. I get all of the water out. It's gone. There's nothing left. So yeah, so I freeze these in liquid nitrogen and then I put them in a freeze dryer, and that takes all of the volatiles and the water out. I extract them. If I do water extracts, that's usually refluxing water, and if it's the organic solvents, then that's usually done with sonication. Yeah. Yes, finely, very finely powdered, and then put in a sonicator. A Hericium species being used in medicines now, or compounds from lion's mane? No, but there is a lot of research on that, and I am working on that side, the medicinal chemistry side of it, with a few people. The problem with them is that they're really fatty, and you can't dissolve them in anything, and so they're not. That's another example of greater the activity side of things, really bad at being a drug. So we're working on that aspect of it. I think that's it. Yeah. Okay. Thank you.